On Handling the Data by M.I. Mayfield. This story appeared in Astounding Science Fiction, September of 1959. Extensive research by Project Gutenberg at Gutenberg.org did not uncover any evidence that the copyright on this publication was renewed. September 16, 1957. Dr. Robert von Engen, editor, Journal of the National Academy of Sciences, Constitution Avenue, Northwest, Washington, D.C. Dear Sir, I am taking the liberty of writing you this letter since I read your published volume, Logical Control, The Computer versus Brain, Silman Memorial Lecture Series 1957, with the hope that you can perhaps offer me some advice, and also publish this letter in the editorial section. Your mathematical viewpoint on the analysis between computing machines and the living human brain, especially the conclusion that the brain operates in part digitally and in part analogically, using its own statistical language involving selection, conditional transfer orders, branching and control sequence points, etc., makes me feel that only you can offer me some information with logical arithmetic depth. The questions raised in this letter are designed principally to reach the embryonic and juvenile scientists, the scientists elect, so to speak. I think the mature scientists are irretrievably lost. For many reasons, some of which will be explained in the following paragraphs, I think that it is of the greatest importance that some stimulatable audience be reached. As yet, the beginners have no rigid scientific biases, and thus may have sufficient curiosity and flexibility about the world in which they live to approach experimentation with a mind devoid of the hierarchy of memory registers which have programmed in erroneous data. What I have to say will not surprise nor shock you, or those who are at present engaged in scientific investigation. In fact, I have read many science fiction stories that deal with the same problem. Perhaps that is the only way that it can be approached, through the medium of a story. Yet why not present it for what it may be? Let me tell it my own way, and then please let me have your coldly logical opinion. As to my background, I am a graduate student in the zoology department of a Midwestern university, working toward a master's degree, or actually a doctorate, we can bypass the MS if we choose, in the field of cellular physiology. My sponsor is an internationally known man in the field. The area of research that I have selected is concerned with the effects of physical and chemical agents on the synthesis of nucleic acids of the cell. Obviously, this is a big field, and I hope to select from among the differing agents one or two that will give positive results. I have been doing active research for about half a year, testing the different agents. As for the fundamental questions raised, I am positive that it would make no difference in what field of science I were to work. By now, I have had enough coursework to realize that when performing any assigned laboratory exercise, they should not be called experiments, even of a cookbook type, little or even major discrepancies arise, and always on the initial trials, no matter how carefully one works. As you are probably aware, the teaching assistant in charge of the lab or the instructor generally runs through the exercises before the class does, in order to get the bugs out of it. I am deliberately generalizing since the above holds for all of the laboratory sciences. So when the student gets confusing or rather contradictory results, the instructor can deftly point out the error in the setup or calculations, or what have you. He may even indicate what results may be expected. The last is critical. Similarly, other students in the laboratory usually have friends who have had the course before and know what results are expected. This technique is frowned upon. Or one may consult textbooks and published papers. This, by the way, is known as library research, and is generally conceded to be indicative of the superior student, especially if he points out the fact that he is so interested that he just had to delve into the literature. By any technique, the expected results are always obtained. Always. And by everyone. The initial confusions that some honest students perpetuate are easily brushed aside as errors due to inexperience, sloppiness, lack of initiative, stupidity of congenital sort, etc., etc. Since being a teaching fellow, even simple cookbook experiments don't seem as cookbookish. Some pretty weird things have happened when I tried out an exercise prior to the class. Fortunately, I was taught to keep data in duplicate, indelible purple hexostick original, and carbon copy. These, vita infra, are a few of such happenings. Elementary General Physiology Laboratory. 1. Initial maximal vagal stimulation. Expected results: inhibition of heartbeat. Obtained results: 1 series of increased heartbeats. Possible explanation: I missed the vagus nerve. 2. Frog nerve muscle preparation. Expected results: a single muscle twitch. Obtained results: a beautiful nerve twitch. Explanation: eyesight? How can nerves twitch? 3. Hypotonic hemolysis. Expected results: red blood cell destruction. Obtained results: crenation. Explanation: switched salt solutions unconsciously? 4. Curarized muscle preparation. Expected results: a synaptic block with no responsive nerve when stimulated. Observed results: a typical strychnine response, violent tetanus, etc. Explanation: again, I switched bottles? 5. I shall avoid the obvious mention of mishaps with mechanical or electrical pieces of equipment. I assure you there were similar deviations in initial attempts. Since I realize that you are preparing a paper on memory registers, stimulation criteria, for the 6th Annual International Meeting of the Society of Theoretical Biomathematicians in London, and are short of time, I shall avoid going into the same kind of detail as above for other biology labs, and get into the real heart of the thing, the research problem. After all, that is what both of us are interested in. By the way, please send me a reprint of the paper when it comes out. I guess I am really hepped up on this, because I've just got to point out for emphasis other incidences, usually of a type that involved missing a whole organ in dissections, or a tissue structure in histology only on the first study, and then rereading the assignment, after knowing what to look for, and subsequently finding it exactly where it is said to be. Ever hunt for an unknown quality or quantity? So it was there all the time? Sloppy technique? Or is this branching at a control point? Cf. LCC versus B, page 251. To get back to my thesis research. The pieces of equipment that I have been using in the research are fairly standard in physiological research. A Beckman spectrophotometer, a Coleman photometer, a Vance-like amino-nitrogen apparatus, a Warburg respirometer, pH meters, Kildahl's, Thunberg's, etc. Mostly I'm in the process of getting used to them. Also there is a high-voltage X-ray generator, UV source, and other equipment for irradiation purposes. We also have an AEC license so that we can get at least microcurie amounts of the usual isotopes for radioautographic work. Now, the literature in my area is pretty controversial. You can appreciate that, especially since Bergbottom at the Kaiser Wilhelm Institute bombarded you with criticisms of your theories. Different, and actually contradictory results have been obtained for the same substance in the same organism. E.g. alkaline phosphatase in the frog liver cell, Monenblick 55, Tripp 56, and Stone 57. To give an example, when I start a run for respiration effects using a Warburg, I don't know what results to expect. Whenever this has been the case, my results have been confusing, to say the least. On nitrogen mustard-treated cells, in some instances, the controls respired significantly more, even with a statistical analysis of variance. In some instances, the experimentals respired significantly more, and in other cases, the respiration for both was exactly the same, even closer than the expected deviations that should be found in any random population. One run, the blank run, containing no cells and grease-free, consumed the greatest amount of oxygen. To cut this letter short, the same inconsistencies apply to other trials that I have made. Whenever I didn't know what to expect, and particularly where the literature was controversial, my results have been completely haywire. Needless to say, I was not happy with this, so I discussed it with other graduate students. They have all encountered the same thing. But most professors won't admit this to be true and merely tell me that my technique is lousy. If anything, I am an overly careful worker. Why is it when I know what results are expected, I get comparable results even on the first trial? Remember, I obtained the expected results when the literature wasn't confused, or when my sponsor, a most important man in my life, gave me a clue as to what kind of results to expect. Only then. Now, this is the heart of the matter. The obvious explanation is the lack of experience, but, and this is what haunts me, what if those so-called contradictory results are meaningful? What if they were executed with care, and they were, and are not the results of sloppiness or inexperience? What if a nerve can twitch? Very respectfully yours, Jonathan Wells. May 3, 1958. Dr. Robert von Engen, editor, Journal of the National Academy of Sciences, Constitution Avenue, Washington, D.C. Dear Dr. von Engen, I would like to thank you for your encouraging letter and advice. I agree completely with your statement that science has a long way to go before we can explain the various inconsistencies that crop up in research. But I certainly can't see how the letter is far too unsophisticated for inclusion in the letters to the editor portion of your journal. While your letter should have calmed me, I feel even more strongly now, after a year of research about the matter, than I did before. I have deliberately postponed answering your letter until I had more facts. I now find that I have accumulated, as you suggested, 3 distinctly conflicting groups of data on nucleic acid synthesis of frog liver cells. 1. There is a conversion of ribonucleic acid to desoxyribonucleic acid. 2. There is a conversion of desoxyribonucleic acid to ribonucleic acid. 3. The synthesis of both types of nucleic acid are independent of each other. In addition, I have some data that I don't want to think about too much that shows that there is absolutely no nucleic acid in the liver cell. Thus, these data, all accumulated in my experimental work, support all 3 hypotheses. Moreover, the literature supports all 3 hypotheses. I intend to go to the Woods Hole, Massachusetts Marine Lab this summer with my sponsor and get some new ideas there, especially since Professor Gould M. Rice from the University of London will be there presenting a seminar series on his work in nucleic acid synthesis in Oryzius. The point is not that there is a conflict in the data, but that the data conflict because there is a conflict in my mind and in the literature. Don't you see it? As you said on page 20 of Logical Control, Computer versus Brain. The order system, this means the problem to be solved. The interaction of the user is communicated to the machine by loading it into the memory. Sincerely yours, Jonathan. August 31st, 1958. Dr. Robert von Engen, Journal of the National Academy of Sciences, Constitution Avenue, Washington, D.C. Dear Dr. Bob, again, many thanks for your letter and encouragement. I especially treasure the inscribed copy of Logical Control, Computer versus Brain, and the current reprint. I am sorry that I didn't get an opportunity to get down to Washington en route to Woods Hole and talk over the whole thing over a bottle of beer. Dark beer. From what I hear of the demands on a first-rate mathematician's time these days, you should be grateful that I didn't get to see you, because I would have monopolized all your time. I appreciate your generosity in extending the invitation as a rain check to me. Your mention of the Duke School of Psychology, my quotes, leaves me cold. It's too obvious and puts the cart before the horse. The important point that I was trying to make dealt not with the possible parapsychological manipulation of equipment or the materials, a la telekinesis, to produce the desired results, but that our science may not be studying natural phenomena and trying to interpret them at all. The point, to get it down in black and white, is that our science, yes, quotes, may be inventing the reality that it is supposedly studying, inventing the atoms, molecules, cell, nuclei, etc., and then describing them, and in the description, giving them reality. While I was at Woods Hole, I had some really good bowl sessions about this very thing. I realize now that I may have been falling into the trap of solipsism, who watches the quad, etc. type of thing. Incidentally, my research is finally beginning to fall into shape. My sponsor and I had some pretty good sessions about it, and some of the screwy results I wrote you began to make sense. I had the good luck to talk to an outstanding man in the field of nucleic acid synthesis, and he was quite enthusiastic about the caliber of our work. He feels quite strongly, but has no real evidence, that the synthesis of both types of nucleic acid are independent of each other, and has pointed out some significant references that I did not know about. I'm anxious to buckle down and really lick this nucleic acid problem in time for a June degree. Cordially, Jonathan. P.S. Please send me a reprint of your lecture on memory banks, transistorized neurons. The lecture was ingenious, but there are some biological phenomena with which I don't agree. Remember, I'm the biologist. Honestly, Doc, don't you think, entre nous, that your idea that a living organism can be compared with automata in picking up informational items and processing them simultaneously in parallel rather than in series is naive? J. October 28th, 1958. Dr. R. von Engen, Journal of the National Academy of Sciences, Washington, D.C. Dear Dr. von Engen, I apologize for not answering your letter sooner. I assume you were pulling my leg when you suggested that I make a science fiction story out of the confused ideas of a beginning graduate student. You might give your idea of a possible science fiction story to one of your acolytes that has some small experience in the field of writing, not science. I am afraid that your other suggestions are not germane to the problem of nucleic acid synthesis and metabolism, a problem that has been occupying all my time. In fact, I've been doing with 3 to 4 hours of sleep these days. With the kind of concentration that I can offer the problem, there is no question that the data are falling into line, and our research is going rather well. We will show, I hope, fairly conclusively that there is little or no interconversion between the two types of nucleic acid synthesis in the cell. Despite your ingenious mathematical approaches for stimulation criteria, in biological research, a very abstruse field, even your multiplex machines with elaborate means of intercommunication are not sophisticated enough, or ever will be, to cope with the complexities inherent in the numerous interacting biosyntheses on the subcellular, ultratopographical level of protoplasm. Sincerely yours, Jonathan Wells. November 8, 1958. To the Editor, Journal of the National Academy of Sciences, Washington, D.C. My dear Professor von Engen, from the tenor of your last letter, it is quite evident that there has been a radical change in your originally sound and inspired ideas, and which clearly indicates to me that a discussion and exchange of basic concept would be fruitless. I'm rather hurt that you question my integrity with the statement about the slick, calculating, career-minded cult of PhD-ism. Moreover, I would appreciate, if possible, the return of my previous correspondence. I don't feel that I am totally inept, for I have been awarded a pre-doctoral fellowship that will support me during the remainder of graduate school. In addition, I'm being seriously considered for a faculty position at an outstanding Eastern university upon completion of my thesis. Should you be interested, we now have an article in press on the Journal of Cellular Physiology entitled Nucleic Acid Synthesis in the Frog Liver Cell, a Definitive Study. We have found substantial evidence which demonstrates that there is no interconversion of the two types of nucleic acid. I cannot help but comment about your recent paper in Scientia. I do not believe that it is at all possible to devise computers which can handle the species of data which we obtain. Your data, being less complex, of course, may fit. Naturally, I have your confidence in the entire matter. Yours very truly, J. Wellington Wells. J. Wellington Wells? Huh. I wonder if that's supposed to be a joke. My name is Joan Wellington Wells. I'm a dealer in magical spells, in blessings and curses and ever-filled purses, in something or other and kells. If you something or other with tax, or to melt an opponent in wax, you need only come down to my shop on the town, number 17, Diggory Axe, or something. I mean, I don't know if it's a joke or not. I mean, they don't use his full name until that point. It's just Jonathan Wells, Jonathan Wells, and then J. Wellington Wells. And I'm like, is that meant to be a joke? It's from The Sorcerer by Gilbert and Sullivan. John Wellington Wells is the sorcerer. That doesn't really have any connection, but it's just a really strange thing. Huh. Sound editing by Barry Howarth.